All right, so thank you for the introduction, and thank you for inviting a hydrogel guy to Nanomedicine Day. Not an invitation I get a lot, but I hope by the end of the talk that I'll convince you that nanoparticles are really interesting building blocks for hydrogels to address some key challenges with using both materials in the context of drug delivery. So really the key aspect of what we've been working on is trying to target mental illness, which accounts for about 7.4% of the world's measurable burden of disease. There's two diseases we particularly were interested in targeting. The first, schizophrenia, affects about 1% of the world's population, and because it affects a lot of younger people, it's actually the fifth leading cause of years lost to disability worldwide, with unexpectedly, a really high economic cost. Parkinson's disease, meanwhile, affects over 10 million people worldwide, and again, a larger than you would expect number of younger people, and again, huge economic cost as a result. Both these diseases do have drug therapies available, but there's two key problems with them. Number one is the drugs typically have very severe side effects, often in terms of weight gain, sexual dysfunction, and other things. And two, we're already dealing with a patient population that has some mental challenges. So patient compliance is a really, really key challenge with this patient population in particular. One of the key reasons that side effects with antipsychotics are so difficult to manage is that it's very hard to deliver antipsychotic drugs or any drug to the brain. With over 95% of the drug typically lost following an oral delivery, which is what is used for the majority of these drugs, due to things like first-pass metabolism, degradation of the gut microbiota, and of course, the blood-brain barrier, which many of you listening probably know much more about than I do. And of course, nanoparticles can address in part some of these blood-brain barrier transport issues by some of the active transport they can do. But there's a pretty limited scope of nanoparticles that do transport through the blood-brain barrier. Even so, the properties that you have to achieve to transport the BBB make circulation and other types of nanoparticle properties a little bit more difficult to achieve often. So in general, through an oral or intravenous delivery, this is a challenging problem. So we've been looking increasingly over the last couple of years using intranasal delivery as an alternative. The intranasal route for drug delivery actually is a combination of three routes, two that go through the trigeminal nerve and one through the olfactory bulb. They all, though, require that we get the drug past the nasal epithelium. So we're replacing one biological barrier, blood-brain barrier, for another, just transport through the nasal epithelium. But the latter is a lot easier to tackle. So keep that in mind as we go forward. If we can get drug from the nose to the brain, though, we can avoid the gut and hepatic first-pass metabolism challenges. It's very easy for patients to administer their own drug through simple sprays, nasal sprays. And because we lose less drug in the process, a higher percentage gets to the brain, we can use lower doses and thus reduce the side effects. The other key aspect of intranasal delivery is the onset of drug activity is actually very, very fast. The consequence, though, is the offset is also faster. So this doesn't really inherently, if we just put a drug itself up the nose, address this patient compliance problem because frequent readministrations are still required. So we asked the question, can we design a delivery vehicle that can be retained in the nasal cavity to address this long-term delivery challenge to reduce the frequency which patients need to self-administer drug, but offers the other key benefits of intranasal delivery? So there are four key design criteria we identified to achieve such a delivery vehicle. Number one, it has to be easily sprayable within the nose by a very broad scope of patient population, from younger people through senior citizens. So we can't require a super thumb to inject. Second, we wanted something that was mucoadhesive, taking advantage of the fact that the nasal cavity is coated with mucus, and there are many approaches to increasing the adhesion between polymeric materials and mucous membranes. Third, we wanted the material to be composed fundamentally of nanoparticles that had good transport properties across the nasal epithelium, so we could get drug to transport to the brain. And fourth, we wanted something that would control the release, both of the nanoparticles themselves and of the drug encapsulated in the nanoparticles. So the solution we came up with is what we call a nanoparticle network hydrogel. And it's composed of two major components, both of which are based on common pharmaceutical excipients that are widely reported. The first is carboxymethylchitosan, which is just a soluble derivative of chitosan, a very well-known, widely used mucoadhesive polymer, with these amine groups on the side as well, which allow us to gel it with other components. And the component we decided to gel it with were starch nanoparticles. And these are based on starch, so that's not too exciting. But the starch that's extruded through our industry partner, Ecosynthetics, through a reactive extruder, to create very, very small particles, 20 to 50 nanometer major fraction particle size, as you see on the graph on the right. They also have a really high water content, And as a result, they're extremely small and deformable, which is what you need to penetrate through tissue, such as across the nasal epithelium. They're also enzymatically degradable, being starch. And because of the extrusion process, they're actually entirely amorphous, which again is a key problem with using starch in other types of delivery vehicles, because a lot of starch is crystalline. What this means is that the hydroxyl groups are very easy to functionalize, which isn't always true for other starch-based formulations. So to make a gel, all we did was lightly oxidize these commercial starch nanoparticles, which are actually produced on the ton scale for things like paper coatings and wood laminate adhesives. We oxidized them to create a few aldehyde groups, which then, when we mixed them with the carboxymethyl and with the amine groups, we could form crosslinks via shift base formation or amine formation. And amines are also highly reversible bonds. So over time, via hydrolysis in the moist nasal cavity, this bond will reverse, and the desired release of these ultra-small starch nanoparticles, we believed or hypothesized, could be achieved. So before we went too far into characterizing how this would work as a drug delivery vehicle, we just wanted to make sure it was practical. So could someone with a reasonably strong thumb actually administer this into their nose through a nasal spray? So what we did is we co-extruded the two precursor polymers with separate barrels of a double barrel syringe through this intranasal mucosal atomization device, which is commercially available, used for other types of intranasal delivery. And what we saw is if we kept the carboxymethyl concentration to 2 weight percent, which again still forms a pretty good gel, as I'll show you shortly, we get a very broad spray area. And indeed, if we look at the difference between spray distance and spray area in the graph below in panel C, you can see that the outer series, where the major droplets can actually spread, it actually, with the 15 centimeter spray distance, which is the average distance to the adult nasal cavity, we can achieve basically an equivalent spray area to the entire area of the nasal cavity. So this is highly sprayable, easy to push, and distributes well across the area we have to hit. If we increase the carboxymethyl concentration, as you can see on the picture on the right, certainly it can be sprayed, but much smaller spray areas are observed because the viscosity is higher. So in one case, we get a very broad, dispersed, droplet-like administration into the nose. The other case, we're basically administering almost a depot into the nose. And we're interested in the impacts of both. So we first, before we actually did the in vivo experiment, wanted to get an idea of how the hydrogel behaves. So in terms of gelation kinetics, when we mix the chitosan and the starch nanoparticles, we could get gelation on the order of a few seconds over to several minutes, so up to about 5 minutes. We particularly favored formulations that gelled in the 2 to 5 minute time range, where it's not going to leak out of your nose just by gravity, but also there's a little bit of time for the formulation to interact with the native mucus layer to interpenetrate and enhance that adhesive bond. In terms of degradation, again, depending on the amount of chitosan and starch you put in, but we see degradation times on the order of 2 to 7 days, which again is fairly consistent with the turnover of native mucus in the nose. So that is appropriate for the application as well. So the hydrogel checks out. So we wanted to know, what could we use this therapeutically? So what we did is first, I'll talk about the drug we chose, which is peyopa. And it's this tripeptide that is a potent dopamine D2 receptor allosteric modulator. This is known in preclinical animal models established by our collaborator to actually prevent or reverse behavioral and biochemical abnormalities associated with schizophrenia. And beyond this being more of an experimental peptide, there's a huge number of peptides now currently under investigation for delivery for mental illness. So we thought this would something we were familiar with in our collaboration, but a good model for other peptides that are under consideration now. And to induce a schizophrenic model, we used MK801, which is non-competitive NMDA receptor antagonist, which has been widely used to model the cognitive deficits associated with schizophrenia, and of particular interest to us, social withdrawal. So what we did is 22 hours before, or immediately before, essentially, we were doing our test, we would administer this via IP, and we get schizophrenia-like behavior for about 2 hours. So if we were looking at later time points, we could just re-administer the MK801, and for 2 hours, we could look at the effect of the drug delivery on the animal behavior. And we did this by taking advantage of the social withdrawal symptom. So what we did is we took 2 rats who had never met each other before, so they weren't co-housed, and put them in an arena, and we tracked their motions with a camera. And essentially, a blinded observer who didn't know what formulation we were administering track the time at which they were under what we called active interactions. So they're sniffing each other, following each other, crawling, grooming, or fighting each other, as rats sometimes do. So by tracking that social interaction time, we hope to see whether those schizophrenic symptoms of social withdrawal could be recovered when we administer our drug. And this is a very complicated graph of the results, so I'll walk you through it a little bit slowly. In panel A, this is without any schizophrenic symptom induction, so this is without MK801. You can see whether we do no treatment with it at all, apply our material alone, or apply the drug alone. We get roughly 2 of the 5 minutes where that we were tracking, where the rats were in active social interactions. We then induce our model in panel B, just using the negative control of artificial cerebral spinal fluid. We see those interaction times are cut to about 1/3 of what they were. If we apply a dose of peyopa intranasally, just alone as a drug, after 1 hour, we see the majority of those symptoms are recovered, but not entirely back to the baseline. But after 24 hours, there's no benefit to administering the drug whatsoever. On the other hand, if we deliver our drug inside our hydrogel, we get complete recovery as quickly as 1 hour. So again, rapid onset of intranasal therapy, and almost complete recovery of the symptoms out past 3 days. And even after 5 days, we're still seeing positive effects. It doesn't completely drop off until about a week. When we increase the concentration of the carboxymethylketosan, so that's more of that bolus as opposed to the general spray that we achieve, there's more of an onset. It's a bit slower to get the drug release started, and we get slower drug release overall, so it ends up lasting about the same amount of time. So it's this 2-8% formulation that we're particularly interested in because it's sprayable and we can get symptom alleviation, essentially complete symptom alleviation over 3 days. To find out what was going on with our starch, to see what was the mechanism of this effective therapy was, we actually labeled these starch nanoparticles with an alexofluor dye and looked at where they were after tissue recovery after sacrifice. What we can see for both formulations, we see even after 3 days, a substantial fluorescence intensity maintained in the nasal cavity. So these are being retained in the nose. And we also see a smaller, but certainly not at all insignificant amount of the starch nanoparticles themselves, not just drug, but the starch nanoparticles in different areas of the brain. So we think what's happening here is there's a combination of active transport of the nanoparticles to the brain through the roots that we mentioned, but also uptake by nasal epithelium cells of the starch nanoparticles, which then allows the drug to be released. It can still itself go through those intranasal pathways and access the brain for therapeutic efficacy. So this works well with the peptides. We asked the question, well, does it work for other types of anti-mental illness drugs? And the one that particularly was important to us to address was levodopa or Ldopa. So it's the current primary treatment for Parkinson's disease. About 40% of Parkinson's patients are on this drug. It's a metabolic precursor of dopamine, but it has extremely low bioavailability, way less than 30% typically. That's a very high-end estimate. There's a lot of gut microbiota degradation, and also because of this catechol functionality on the drug itself, it's extremely prone to oxidation in the physiological environment. So patients that are on this drug, which many are on it for the rest of their lives, high doses are required, increasingly high doses over time. Patients often experience these off periods where at some point it just doesn't work anymore. So we're interested to see whether we could modify and take advantage of the fact that these starch nanoparticles are amorphous, so they're easy to modify with affinity groups for different types of drugs. Could we modify this sprayable hydrogel platform to deliver Ldopa? So what we did is we put our aldehyde groups in by the oxidation reaction, just like we did before. But before that, we conjugated a phenylboronic acid group on. And phenylboronic acid groups are well known to interact through reversible covalent bonds with cis-diol groups, such as the catechol group on Ldopa. So through this reversible covalent interaction, we thought we could both stabilize the drug versus oxidation and also control its release over time to get longer term activity following a single administration. So we formulated our hydrogels the exact same way. So carboxymethylketosan in one barrel, our starch nanoparticles in the other barrel with the drug, co-extrude it through the nebulizer. And what we saw was dramatic differences in the drug stability we could achieve. So if you put levodopa for 48 hours in PBS, you see the solution turns from clear to essentially entirely black. This is indicative of the oxidation, which actually produces a toxic byproduct, as I'll show you in a second. If we use our conventional gel that we use for the peptides, we get much better behavior, but still some brown, so there's still oxidation going on. If we lower the pH just a little bit using MES buffer to 6, so again, something that the nose can tolerate, but a little bit more acidic, you get a bit better stabilization. If we then use our phenylboronic acid modified delivery vehicle at pH 6, you can see the solution is almost entirely still clear. So we can almost entirely suppress this undesirable oxidation reaction. So this is hugely beneficial for stabilizing the drug. We then wanted to test whether the biological activity was also maintained. So to do that, we actually used a C. elegans electrotaxis model, where we first exposed the C. elegans to a high dose of 6-hydroxydopamine, which is a well-known induction agent for Parkinsonian symptoms in worms, but also other animal models. And when we took those worms and incubated them in the presence of our gels, in the presence of bacteria as well, so the worms had a food source. At different periods in time, we took one worm out and placed it in a microfluidic electrotaxis chamber. So C. elegans, when applied in an electric field, will swim in the direction of that electric field. So if we can confine that in a microfluidic chamber, we can track the rate at which they swim, which can relate back to the efficacy of the drug formulation. So I'm going to start these videos. So the blank control on the right is after the C. elegans is exposed to the 6-OHDA, but without any treatment. And you can see really uncoordinated swimming and much slower swimming. And this is indicative of a Parkinsonian-like model. On the other hand, on the left, when we expose those same worms to our gel, we see much faster swimming and much more coordinated swimming, typical of a worm that is not treated with the Parkinsonian induction material. You can see that in the average speed measurements. So with just free Ldopa, we get some short-term recovery, but then it drops off, and indeed the worms die after 24 hours due to the toxicity of the oxidative byproduct. Whereas if we do this in the presence of our gel, we can deliver the drug over 48 hours and get complete speed recovery. We can even deliver 3 times more drug and not cause any toxicity in the worms than we would achieve with the drug alone. So we're really excited about this, and we're going to rat studies this fall to see if this works on a larger animal. So in conclusion, I hope I've convinced you that nanoparticles are really interesting building blocks for hydrogels, because you can essentially take advantage of the properties of both materials. So the bulk nature of hydrogels, their ability to control the release of nanoparticles over time, as well as their ability to adhere to things like mucosal surfaces, while retaining the beneficial properties in terms of penetration and drug carrying, ability to tune the nanoparticle affinity for different drugs of the nanoparticles themselves. So I'd like to thank the students that did this work, particularly Michael, Ali, Andrew, and Xiaoyun, as well as our collaborators Ram Mishra and Begwati Gupta at McMaster, and our funding partners. Before we wrap up, I just want to do a 30-second pitch for an NSERC Create program that we have just started at McMaster called the NSERC Create Training Program for Controlled Release Leaders, which is really an effort, a collaborative effort to train graduate students and postdocs in controlled release broadly, both for drug delivery as well as agriculture and food science, using a partnership model with universities, industry partners, and nonprofit organizations like the Canadian chapter of the Controlled Release Society. So if this is interesting to you, we are recruiting students now. Please check out our website or follow us on Twitter. Thank you.
Thank you, Todd. So maybe quick answers for maybe 2 questions. First one, did the animals feel any respiratory discomfort due to intranasal delivery?
That's a great question. So that is something we did track because we were concerned about, the very narrow cavity actually being blocked by the hydrogel. We saw no evidence of that whatsoever, and we would do repeated readministrations on the same rats just to check that. So we would readminister every day over several days just to see if there was a buildup of gel, whether that would cause respiratory distress. And we saw no evidence of behavioral changes whatsoever. We believe this is just because we're getting very, very thin layers of gels that are attaching to the surface of the mucus, and also some penetration of those gels into the mucus because we chose the slower gelling time material. So we don't see a sort of the blockage and the respiratory symptoms that you might be concerned about.
Okay, another one, any toxic byproducts?
Of the delivery vehicle?
Yes.
No, so it's basically chitosan and starch. So these are both, things that can be enzymatically or oxidatively degraded and have been used commonly for other types of drug excipient applications. So we saw no evidence of any sort of toxicity of the degradation products.
Okay, thank you very much, Todd. So we still have some unanswered questions in the Q&A. Please do that online. Thank you so much.
I will. Thank you.